These are some of the organisms that can do photosynthesis. These are called autotrophs because they make their own food, and the photo indicates that they use light as a source of energy to do so. We have a familiar forest here on the bottom left, but the top picture shows a kelp forest made of seaweed, which are not plants, but belong to the eukaryotic kingdom of protists. On the right is cyanobacteria, which, despite its name, is also a protist. It was reclassified after naming. Oops. The eukaryotic organelle used to do photosynthesis is the chloroplast, present in leaf cells of a plant. Don't forget that these cells, they also have mitochondria, right? Chloroplasts, they're not one-stop shopping. They don't do it all. Mitochondria is still needed to generate ATP. It's important to think of where each part of photosynthesis is occurring in the chloroplast of the cell. The chloroplast, similar to the mitochondria, has an outer and an inner membrane. However, there is a third membrane inside that is called the thylakoid membrane. It surrounds what looks like these green stacks of coins called granum, or plural grana. Remember, this is a chloroplast, not a cell, right? This is one of many organelles contained in a eukaryotic cell, like the plant cell here on the left. Talking about light is like walking the border of what I understand about physics. But I have this simplistic understanding of it, so I think sometimes that makes it easy for me to get some of the basic concepts across. All right, so light is an expression of energy. That energy can be measured in wavelengths. Up top of this diagram here, we have a scale of wavelengths from really tiny nanometers on the left to kind of big meters on the right. The shorter the wavelengths, the more energy they have, like gamma rays and X-rays, right? That's why they cause cancer. The longer the wavelengths, the less energy they have, like radio waves and microwaves, but that does not mean that you can stand next to your microwave. Light that is visible to the puny little human eye is only a very small portion of the scale, right? See, ranging from 380 to 750 nanometers. These wavelengths of visible light can be either absorbed or reflected by pigments like chlorophyll. We see chlorophyll as green, at least if you're not colorblind. That's a whole different PowerPoint. The green color is the wavelength that is reflected or transmitted by chlorophyll. The other wavelengths or colors are absorbed by chlorophyll. If you want to think in terms of color instead of wavelength, go for it. This is not physics. It's fine for this class. Check out the graph on the left. It's a graph of the wavelengths or colors that are absorbed by the different chlorophyll pigments. And there's chlorophyll A and B. Notice how there is very low absorption for the blue-green wavelengths. They reflect that. That's what we see. But there's lots of absorption for the red, orange, yellow, and purple wavelengths. They absorb that. We don't see that. Yeah, but plants, they're not limited to these colors, right? They don't lose out on the blue-green wavelength. I mean, they have energy, the blue-green wavelength, and we want to be able to use it. So there are additional pigments in plants like carotenes, which are obviously orange, and xanthophylls, which are yellows and red. We see these pigments when the chlorophylls degrade in the fall before the leaves drop. For example, xanthophylls absorb the red, orange, yellow wavelength, but not the blue-green wavelength. So plants that have both these pigments can absorb almost the entire spectrum, expanding their available energy from the sun. Okay, now that we have a grip on light energy and how we measure it in wavelengths, let's look a little deeper at the energy it provides. We designate the term photons to light energy. Think of a photon this way, as a little packet of light that comes in from the sun, a photon. it can excite, and thus it can pass on the light energy to electrons. Electrons in chlorophyll, they actually skip to another shell, holding the energy from the photon. We do something similar to this when we use these glow-in-the-dark necklaces that you get at carnivals. By cracking the necklace or breaking it open, you excite electrons in the substance, and the excited electron emits light energy. Over time, the electron kind of fizzles out, emitting less and less energy. You can delay this process by putting the necklace in the freezer. Plants, instead of letting the electrons fizzle out, they harness that energy into something usable for biological systems. Okay. With an understanding of how the pigments in plants can absorb light energy and transfer it to an electron, we start to think of cellular respiration, right? Where the energy from an excited electron was harnessed with an electron transport chain. We're going to do the same thing here. We're going to use the same machinery, ATP synthase, electron transport chain. We're going to use that to harness the energy from an electron excited by a photon. I like this overview diagram of photosynthesis. Check out the background of the diagram. It's an outline of a chloroplast. You can see the outer membrane and the inner membrane, and then you can see those thylakoids that had what looked like stacks of coins. There are two main steps to photosynthesis. There's the light reactions and carbon fixation. I like that this diagram has the light reactions on the left and the carbon fixation on the right. Take a moment to look at the cycling of molecules that we have between the two steps. It seems that the light reactions make ATP and NADPH, which then is used by carbon fixation. You might be wondering what NADPH is. So let's just keep it simple. It's just another electron taxi, right? However, this electron taxi is specific to chloroplasts, whereas NADH is specific to mitochondria. Take a look at the other things associated with the light reactions on the left, like light, water going in, and oxygen. In carbon fixation, we seem to have an input of carbon dioxide, which we know that plants require. And then we have an output of a molecule called G3P, which, let's just keep it short and sweet, it's a sugar. So when you start learning about photosynthesis, you're kind of told that glucose is the final product of it. And it is, ultimately it is. But in the short run, carbon fixation, that process makes G3P. G3P, or glyceraldehyde 3-phosphate, is a very versatile molecule. And it can be used for, so many purposes. Of course, G3P can be used in cellular respiration, but it can also be used to make starch, cellulose, and other organic molecules that are used by plants. Oh, what is this? I'm kidding. Remember, when you look at a diagram like this, you first want to identify the features that you know. Strange. Some things here seem somewhat familiar. Let's start with location. The membrane, coiled membrane that you see here is a thylakoid membrane. It surrounds those stacks of coins called grana in a chloroplast. The thylakoid membrane separates the inner thylakoid space, like the inside of a coin, from the stroma of the chloroplast, which is just kind of like what's in the inner membrane. We're going to build a proton gradient into this thylakoid space, just like we built one in the intermembrane space in cellular respiration. At the top right-hand side of this diagram, we have an electron taxi, but this one has the name of NADPH. You know what? Don't worry about it. Just pretend that the P means photosynthesis or something like that. It's fine. It's fine. At the bottom, we have ATP synthase. Right? It doesn't work any differently than it did in the mitochondria. It's the same thing. It's going to allow the protons to diffuse, turn the rotor, and then make ATP. We have an electron transport chain in there. Did you see it? Note that it is moving protons from the stroma into that thylakoid space. Flanking the electron transport chain are two things called photosystems, and those are new to us in these diagrams. Photosystems are quite complicated, but we can reduce them to basically a complex that contains the pigment that makes the thylakoid membrane green. We have photosystem 1 and 2. Photosystem 2 will be used first, and then photosystem 1 will be used second. I'm sorry. They're named due to their discovery. So some person found photosystem 1 and was like, yes, here's the thing that does photosynthesis. And then they found photosystem 2 and they were like, damn it. So they're not named for the order in which they're used. They're named for the order in which they were discovered. Look how the electron transport chain is between the photosystems. All right. So finally, let's locate water that we know plants require for photosynthesis. Right, we see it here on the left, and it seems to be donating 2 electrons to this photosystem. By donating these electrons, water is oxidized to molecular oxygen. And that seems right to us. We know plants emit oxygen. Oh, I'm sorry. One more thing. The photons. This is the light reactions, right? And we need those little packets of energy to get photosynthesis, the light reactions going. Okay, let's put this in motion. A photon comes into photosystem 2. See it here over on the left? It excites an electron. That electron leaves the photosystem to the electron transport chain. Right? It does that hot potato, hop, hop, hop. Those electrons that are lost, apparently it looks like they're being replaced by water. All right. Okay. As the electrons hop through the electron transport chain, protons are pumped from the stroma to the thylakoid space, right? And as the electrons hit each protein, they lose their energy as they hop from protein to protein. But this time, there's nobody to accept the electron from the electron transport chain. So work with me here for a moment, because this next process, it kind of happens at the same time as the one I just described. Another photon at the same time, somewhere in the city, in another location, a photon enters the pigment in photosystem 2, exciting the electrons. These electrons are loaded into an electron taxi, NADPH. These electrons will be shuttled to the stroma to be used in carbon fixation, and that's the second step of photosynthesis. The electrons from the electron transport chain, okay, so they replace those ones that were lost to the taxi. So all this time, really what we're doing here is we're just shuffling electrons from photosystem to photosystem using electron transport chain, right? And we're pumping protons to the thylakoid space while we're doing it. That's all we're doing. That's it, right? We're moving electrons and pumping protons. These protons, they build up in the thylakoid space and they diffuse to the stroma through ATP synthase, which will harness the energy of that movement into the bond of ATP. Okay, so that was the light reactions. Let's move on to what's called carbon fixation. Sometimes this is also called the dark reactions or the Calvin cycle. Carbon fixation, it takes place with enzymes in the stroma of the chloroplast. Take a look at the inputs on the diagram from the right, on the left. I like this diagram, it's nice and simple. The inputs for carbon fixation are the products of the light reactions, ATP and NADPH, as well as carbon dioxide. The carbon dioxide will enter the cells of the leaves via these things called stoma, which you can see here in the picture on the right. Stoma, which is singular, stomata, which is plural. Stomata are like little mouths on the bottom side of the leaves that, when open, allow the exchange of gases into and out of the leaves. Carbon fixation is as complicated as the citric acid cycle of cellular respiration. It is, it looks just like it, right? I think you've gotten the message that I want you to focus on inputs and outputs with reactions like these more than the specific steps. Check out how carbon dioxide enters this cycle. It's accepted by an enzyme called Rubisco. I'm sorry, I just like that name because it makes me think of Nestle Crunch that's made by Nabisco. All right, by using ATP and NADPH, G3P is made by way of reduction, the gain of electrons. But check out how intense this cycle is. There are 5 G3P molecules already and always stuck in this cycle. We make only 1 G3P by way of reduction. But then we spend some ATP to recycle the 5 G3P into this molecule called RUBP, which is basically a sugar. RUBP is it's a 5-carbon sugar to which carbon dioxides will be attached to start the cycle again. I mean, why do you need to know so much intense information about the 2 steps of photosynthesis? Why can't we just leave it at plants are green, they make sugars, what else do you really want to know? Oh, they make oxygen too. I don't know, that's kind of important. All right, so you need to know enough to pass the quiz in this module, but this knowledge also lets us understand plants, like the pineapple plant. It blooms this flower over here on the right that's known as a multiple. You can see the purple flowers coming out of each segment. Each of those flowers will be pollinated, and then they'll fuse together to become a pineapple fruit. You can purchase pineapple plants at local home improvement stores and greenhouses. They're really common and they're really hardy houseplants. One thing about them is that they thrive in desert environments. In fact, they have a specific adaptation to dry climates, as they do the light reactions during the day and they store the ATP and NADPH. And then they open their stomata at night and do the dark reactions, letting go of the oxygen and taking in carbon dioxide. The daytime in the desert is so dry that opening those little mouths, those stomata, would desiccate the leaves and it would dehydrate the entire plant. By opening the stomata only at night, gases can be exchanged. Since carbon dioxide, it's not needed for the light reactions, but it's only needed for carbon fixation. And this gives these plants an advantage in the dry, arid climates of deserts and other subtropical regions.